Much and sorry for all the technical issues. I'm very happy to be with you here today. So I'm going to present a little bit of my PhD research project at McGill University, which is titled Comparative Genomic Analysis of Primary Medulloblastoma and Leptomeningeal Metastasis. So just to give a little bit of introduction, medulloblastoma is one of the most common malignant pediatric brain tumors. It's we have 250 cases per year, so which is like almost 20% of all the pediatric brain tumors, and usually it arises in the cerebellum. But interesting thing about medulloblastoma is that it has a very complex heterogeneity. So far, transcriptional analysis and genomic analysis have identified at least four molecular subgroups, but more recent research has have identified at least another 12 subtypes in these four groups. Now, and each subgroup will be affiliated with the survival an outcome because there will be groups that are more they will have more tendency to develop metastasis. Now, in the context of metastasis and medulloblastoma, this is called as leptomeningeal disease because it's the spread of metastatic cell along the brain and spinal cord and the meninges, and it's an universal predictor of poor outcome. And within these groups, usually group 3 is one of the most aggressive. Almost 50% of the patients that have group 3 medulloblastoma will develop leptomeningeal disease, which is also, to abbreviation, we also call it as LMD. And the survival is less around is maximum could 5 years. So it's for us, it's as a challenge to understand these type of tumors, also because we have the lack of samples. So, and because of this, it's still incurable, and no progress has been made in the last 30 years. And as I said, is a metastasis compartment is a very understudent field because of the lack of samples, and so we need new approaches to try to develop platforms to better understand this disease, so we can formulate hypotheses and identify in the future targets that are specific for leptomeningeal disease that can help us to prevent and treat it. So for this, our hypothesis is that we believe that there is indeed difference between the primary medulloblastoma and metastatic medulloblastoma. So we aim to understand a little bit of the molecular events that drive the subclones of the primary to metastasis, and so having this will help us to offer therapeutic targets for this. So our aims is like to develop, as I said, because of the lack of samples, it's a real issue. We need to find new strategies. And one of these is like to develop metastatic and primary patient-derived xenograft models that faithfully recapitulate primary and leptomeningeal disease medulloblastoma. And having these compartments, we can perform genome-wide molecular profiling to see the differences between the two compartments. And so once we identify targets, we aim to identify and genetically validate, so we can they can be laterally tested for further preclinical models. So to establish the primary medulloblastoma models, we work with commercially available PDXs. So they, as you can see here, they are different types of PDXs available for each of the molecular subgroups. So which I have highlighted is the ones that we have worked on. So it works just with an orthotopic transplantation. It means an injection at the site where the tumor develops, and we wait, as you can see in the image, the appearance of the tumor, and we monitor through the observation of neurological symptoms in the mice. And then we do microdissections and cell dissociation sequencing, and then it's ready for bioinformatic analysis. Now, for the metastatic LMD models, it's a little bit a different approach. So we work based on the premise that metastatic cells usually leave it was recently discovered that metastatic cells leave the central nervous system, travel through blood, but usually, or very interestingly, they go back and they attach to the meninges to form metastasis. So to have this in mind, it was developed this approach. So tumor cells are injected in the flank, and then we wait for the growth of primary tumor, which is removed. So that's to give more time for the cells to go and travel to the central nervous system and develop LND, until we have, as you can see here in the image, until we have the appearance of metastatic medulloblastoma. And we follow the same approach as the primary, this microdissection, cell dissociation, sequencing, and bioinformatic analysis. The idea is to have these two compartments and do differential expression analysis. So, but before doing an transcription analysis, it was worth for us to validate the nature of our PDXs. So we know that these PDXs, as I say, they were commercially available. So there is a lot of mutations that have been reported. And to validate that the faithfulness of these PDXs, we develop a bioinformatica pipeline to identify short variants on indels from the RNA-seq. And so we could identify the same mutations that were reported from these PDXs. So we could also to validate that we were working with these models, because it happened to us that we thought that we were working with one model, but when we checked the mutations, it was another model of medulloblastoma. So that helped us like to refine and to be sure that the PDXs that we were working were faithful. And now that we have this, now that we validated, we perform differential expression analysis. For this, I would like to highlight two important points. We use Xenom. It's an algorithm that helps us to classify human reads from mouse, because one issue that we always deal with is like, because we have the tumors growing in the mouse, when we are taking the samples, we are at the risk of taking mouse reads, and that could bias our analysis. So what we do is like we try to separate and to isolate only the human compartment, so we can work based on that. And so at the end, we have these clean files that only represents the human compartment. And then also we use special for the quantification Salmon, which is allows to do the analysis independent of alignment. And it's very nice because it can compute the TPM, so transcripts per million, right away. So in our lab, it's easy to work with this. And so as you can see the results here very quickly, this is as the PCA. You will see three different models of group 3 medulloblastoma, which is called MMB, MED14, and MED411. And each, as you can see, has the match primary and meds. And you can see there is a very nice clustering between primary and meds. So that's for us is we see that primary and metastatic are indeed transcriptional different, close and separated together from the primary, as you can see here specifically for the one of the PDXs models, and also in the heat map. And also doing the gene set enrichment analysis, we also see that there are overrepresented pathways in the metastatic compartment rather in the primary. And it's worth to note that one of the pathways was oxidative phosphorylation, which was already in a previous paper was identified that the OXPHOS or metabolic phenotype is involved with the leptomeningeal disease progression in medulloblastoma. So it's was reassuring to find like these pathways have something to do with the metabolism and survival and the spread of metastatic cells along the leptomeninges. And doing a comparison between these, also we analyze all the differential expressed genes. And across the models, we found a total of 80 genes in common, differential expressed among all the models of group 3. But interestingly, we only found one just in common, only in common among the whole 3. And this gene was FGBP. And as you also, we do some of histological stainings and in the areas of the tumor, and you can see a little bit the expression of FCGP in these areas. And right now it's unknown the function of FCBP, but we know it's a glycoprotein that has been found differential expression in a different types of tumors, such as ovarian cancer, also in metastatic colorectal carcinoma, and also glioma. But in medulloblastoma, it has not been reported before, so that's for us it's exciting. And also because we search through another data sets, for example, in human data sets, and also from GeoOmnibus. And for example, this, as you can see in the first one, it's from a collaborator from the University of Toronto, Dr. Michael Taylor. Um, um, it's human data sets, and you can see FCBP is expressed in the metastatic compartment. And also this is in your left, no, sorry, in your right, there is also one of the first studies between primary and medulloblastoma was microarray data. Also you can see that the FCBP is overexpressed. And what recently, now that we have these new technologies of single cell sequencing, it was one of the few studies, because medulloblastoma is a very rare tumor, one of the few studies where they have developed a single atlas of human leptomeningeal metastases. They profile patients with a primary lung and breast cancer with a metastasis in the meninges, so with LMD. And we can see in the circulatory tumor cells high expression of FCGP. Um, so that's all this data. It seems like this gene has something to do with the development and progression, but we are still working of how it works. And just for conclusion and future perspectives, we can conclude that indeed primary and leptomeningeal disease are transcriptionally different. They are signaling pathways more enriched, and here we are interested to develop more approaches to study what is the role of the oxidative phosphorylation metabolic phenotype. And we see that FCBP is a potential candidate to validate its role in the development of LMD in vivo. So right now we are working to develop a CRISPR system activator to overexpress FCGBP, and to inject and to transform to modify cells that we know that they don't is a medulloblastoma cell line, but we don't know that it's not they are poor in metastatic development. So we, with this approach, we aim to if we overexpress FCGBP, we will check how is the increase of leptomeningeal disease in mice. And also we are also working to test the ordered candidate genes, because there is I mentioned there is 80 genes that we have found so far, but 79 is remaining. So also we want to validate these genes, so hopefully we can identify or have a list of candidate targets to keep tracking if these genes of these molecular events are targets to treat or prevent metastatic medulloblastoma. So thank you so much for your attention. Apologies again for the technical difficulties, and I will be happy to take any questions.